Hey guys, today we're going to be talking a little bit about the urease test that we're going to be doing for our lab. So when amino acids are broken down by an organism, a common byproduct to that is urea, and certain microorganisms then have an enzyme called urease, which can break urea down even further into carbon dioxide and ammonia. So kind of with that in mind, the way that our test tubes are set up to test for urease activity is that they have a bit of urea in them, and then they have a phenol red pH indicator in them. Our phenol red pH indicator has this kind of peachy yellow color from about pH 6.6 to about pH 8, where it changes to more of a pink color. So if an organism has the ability to break down the urea and produce ammonia, which is an alkaline product, it's going to increase the pH of our tube, eventually turning it pink. So when you go through and you inoculate your tubes, again, they should start out in this sort of peachy color, and then after incubation times, when you come back to look at them, if it stays peachy, it's a negative result for urease, but if it turns to this kind of pink color, then it is a positive result. And in terms of actually inoculating our urease tubes, you can inoculate them really with a standard broth-to-broth transfer, that's fine. If you have your sample on a slant or a plate, you can transfer from the slant or plate into the tube as well. No big deal. But that should really cover what we need to know for the urea.